Hello students, today we discuss about the continuation of molecular basis of inheritance. In that one, another subtopic that regulation of gene expression. In this, normally gene expression results in formation of peptide. In that one, this gene expression, how many types of this regulation of this gene expression it must be takes place under different types of. First of all, what is meant by gene? That means gene itself express a particular phenotype characteristic features of that particular organism. That is, so there are different types of genes are present, like cancerous oncogenic genes, or apart from this one, overlapping genes, as well as initiation gene, termination gene. Then apart from this one, constituent, non-constituent. Apart from this one, related to plants also NIF, nitrogen fixation gene. Like that, many, that means multiple or overlapping such type of or pseudo type of many different types of genes are present. So according to this particular topic, we discuss about different types of, among these one, what type of genes which regulate that particular protein synthesis and all, that is related to constituent genes, that means non-regulated genes or housekeeping genes. Non-regulated or housekeeping genes, so it always express for cellular activities. For example, glycolysis as well as ATPase, it always expresses. Then non-constituent otherwise we call it as regulator genes or luxury genes, whenever cell required, whenever the type of the cell, whenever necessary in that particular action, in that particular place, then only in time to time it express, not continuously, whenever needed, whenever necessary, in that particular necessary of the time only, the gene it must expresses. That is we call as non-constituent genes and all. Here, to study about gene expression, so related to biochemically, what it is called as, that means mechanism of gene expression, scientifically known as related to, that is biochemical, related to genetics and all. So biochemical genetics. So mechanism of gene expression is known as biochemical, that genetics, otherwise it is known as. Here they must give different types of, among these one, non-constituent or regulated genes they must be given here. According to our particular topic, regulated genes or switch off genes, otherwise it is called as switch on or switch off genes and all. In this one, we discuss about here what is meant by when it is called as, that is how many types means related to many multiple types are there. According to our topic, there is a constituent as well as non-constituent. Then how many regulation how many types of regulatory genes are present. One is the repressor gene. According to regulation, how many types of regulatory genes? One among the regulatory gene, that is the repressor gene. Another one, inducible gene. Repressor, it always express, it always bind, does not take place that particular actions and all. So to stop the particular actions, particular transcription, translation process, but it takes place continuously, except that particular deviation. Then inducible, with the help of inducible, with the help of activator, then only it function, then only it activated, then only it continues as transcription, translation process and all. That is inducible and all. It is depends upon that particular type of inducer. That's why it is called as inducible genes and all. Then these particular expression of the genes, how many different levels are present in prokaryote? There is no particular levels in prokaryotic. Why? Because it is present only initiator as well as termination. Only two it is present. But how many different levels present in eukaryotic organism? Levels in eukaryotic one, that is transcriptional level. It is present transcriptional level. That means initiated transcription. Nothing but heterogeneous nuclear RNA. Then apart from this one, processing level. Processing level means that particular splicing or splitting of genes or splicing, regulation of splicing, that exons, introns, that particular area and all. Then transfer of mRNA from nucleus to cytoplasm. From nucleus to cytoplasm. That is also one level and all. Then translation level. So these different levels are present in eukaryotic organism, whereas prokaryotic initiation, termination only is present. Then apart from this one, in that prokaryotic organism, a specific gene regulate different metabolic. Why? Because regulation of the gene expression, it must control based on metabolic as well as chemical. Apart from this one, environmental factors. So regulation of gene control under metabolic, chemical, along with this one, the environmental factors act as an important role. Then here different types of operon models are present. Why? Because the controlling of the protein synthesis was first elucidated by Jacob and Monod and all. Here, Jacob and Monod. So in 1961, these two scientists explained related to the control of protein synthesis that was elucidated. That is related to Jacob and Monod described how it must control the protein synthesis, especially in prokaryotic one. Why? Because there is no particular such type of, it is present related to eukaryotic and all. Here, especially in prokaryotic one, how it must be regulated and all. That is related to, that is operon model. They explained about the operon model. In that one, different operon models present in only prokaryotes. So statement level of questions, that is very important. Operon model system present only in prokaryotic organisms, not in eukaryotic organisms and all. In operon, there is lac operon is there, many different types are there. Lac operon as well as tryptophan operon, valine operon, histidine operon. Arabidos, operon, like that, as well as arginine operon. Many different types are present. In this one, it will be regulated different types of gene. Operon means a group of. Why? Because here one can consider our very important role under different types of genes. Particular gene present on that area we call as histron. Here, polycistronic area. Why? Because specific mRNA present many number of the genes and all. That's why polycistronic and all. Polycistronic. In lack operon. Why? Because in operon group of genes arranged and all. In that one, how many number of structural genes are present? 3 structural genes are present. In that lac operon, 3 structural genes are present. Whereas tryptophan operon, 5 are present. Whereas valine, it is also 3. Whereas histidine, it is present 9. Whereas arabidose, it is also 3. Whereas arginine, it is also present in 5. But here, in histidine, 9. Tryptophan 5. Remaining, arginine also is present 5. Remaining, rest of these 3 structural genes. Why? Because structural genes act as a vital role to transcribe mRNA as well as formation of polypeptide and all. These are structural genes and all. In this one, in this particular operon model, that Jacob monad and all, here lac operon model and all. In that lac operon model, what originally this lac operon is switch on or switch off. Originally it is the switch on. Actually, that lac operon, originally it is the switch off and on. Because of that particular protein, it must be synthesized. Before that one, what are the genes present in operon and all? What are the genes are present? That is regulator gene. Regulator gene is present. Regulator gene helps synthesis of biochemical, that is one particular protein. It acts as, if it acts as repressor, it stops that further action, the further metabolic activities like transcription, translation, that means formation of polypeptides and all. Similarly, if it synthesizes that particular inducer as a protein, that is, it automatically, the metabolic activities, it takes place continuously and all. So, what is the important role of regulator gene? To synthesize protein, whether it is repressor or inducer. It depends upon switched on or switched off. Why? Because these gene, whatever secreted, it binds into the operator gene. Then promoter gene, here simply read to synthesize protein. Either it is repressor or inducer. If it is repressor, one can stop further reaction. If it is inducer, one can continuation that particular action and all. Then promoter gene, it helps to bind RNA polymerase. To bind. Why? Because RNA polymerase acts as a crucial role for transcription process and all. Then operator. This operator gene. What is the important role of operator gene? Operator gene helps, if it is bind, it depends upon regulator gene. Regulator gene, after synthesis repressor, if it bind operator, it stops the further structural gene function. So it control to operate structural genes and all. To control structural genes. Then structural gene helps to transcribe as well as translation. It helps in transcription of mRNA as well as translation of proteins. Both transcription and translation, if it is activated, if it is actively present in that specific operon and all. Suppose here in that lac operon, if it is lac operon, DNA molecule. In this DNA molecule, it is DNA molecule. In this one, it contains the promoter as well as inducer, otherwise it called as regulator gene. Apart from this one more promoter gene, operator gene, then Z, Y, A. These are structural genes. These are structural genes are present. If inducer is absent, whenever this condition inducer is absent, whenever inducer is absent, then what is the going on and all? Then in this regulator it is synthesized. Why? Because in that specific that promoter gene, one can combine, one can add in that RNA polymerase. Due to adding of RNA polymerase, it transcribe and formation of repressor RNA. This is repressor mRNA. This is the transcription. Then in translation, repressor protein formed. Repressor protein. This repressor protein, it bind with the operator. So the repressor protein bind with the operator gene. Whenever operator gene to stop for binding of RNA polymerase in the promoter region, then to stop binding of RNA polymerase. Whenever to stop RNA polymerase on the promoter gene, then the further transcription translation also stopped and down. Then further transcription and translation process also stopped and down. So automatically it must be stops everything and all. If inducer will be present, this you remember carefully. If inducer, when it becomes switched on, when it becomes switched off and on, promoter, that means related to repressor. So repressor protein plus operator, it becomes switched off. Switched off this one. Whenever repressor protein plus inducer, then it is switched on. One is switched off, one is switched on and on. This is switched off condition. Then similarly, the same if it is present, whenever combined with one inducer, whenever combined with inducer, whenever combined with inducer unable to bind on operator, then automatically the RNA polymerase bind on promoter gene, then further process takes place. That means structural gene. This structural gene transcription process to form inducer mRNA. That means here, if inducer is present, here another very important exam point of view. Which one act as the inducer? That is lactose and all. Inducer, that is lactose. That's why it is called as lactoperon, lactose operon and all. Because of three structural genes, it regulates and synthesis of the proteins. Here, proteins means in the form of enzymes, it must be produces and all. Suppose G, that Z particular gene is there, it synthesizes beta galactosidase, that is beta galactosidase enzyme. Then Y, it translate permease, whereas A trans acetylase. So this translation process, it takes place and all. Here, what is the important role? Here, the beta galactosidase act as a crucial role. Whenever sucrose, whenever that lactose is present, the lactose, it break down into two monosaccharides and all, as well as glucose as well as galactose and all, with the help of beta galactosidase enzyme and all. Then what is the use of permease? Permease helpful to enter towards that cell membrane and all. Permease supporting to entering of beta galactosidase into the cell. Then trans acetylase, the acetyl group entering into the beta galactosidase. So overall, all these enzymes helpful for completion of that disaccharide converts into the monosaccharides and all. How much period of time it is present, how much time in that particular culture? Why? Because in that all the bacteria, if it add into the lactose, then automatically one can synthesize all these different types of enzymes and all. If absence of lactose, automatically unable to synthesize these enzymes and all. But this enzyme helpful for breakdown. That is important. What is the specific enzyme helpful to break down lactose into two monosaccharides like related to glucose as well as galactose and all. That is important. Now, tryptophan repressor operon and all. Tryptophan repressor operon. It is also observed that the Jacob Monod scientists only. Here, 5 structural genes are present. Normally, this protein, whenever synthesized, then automatically it represses and all, more than normal amount and all. So, normal tryptophan operon, normally it is switched on. Normally it is switched on. Whenever binds tryptophan, then it is switched on. Very important. Lactose operon, normally it is switched off. Whenever inducer added, then it is switched on. Tryptophan operon, normally it is switched on. Whenever that respectable of repressor enter, then it is switched off and on. Why? Because here two repressors are present, aporepressor, corepressor. Whenever aporepressor combined with corepressor, then it unable to process, it unable to transcribe, it is inactivated that structural genes and all. Here, in this DNA molecule related to promoter as well as regulator, along with this one, promoter, operator, then L, then this is later attenuated, then it follows structural genes. Here 5 structural genes are there like E, D, C, B, A and all. These 5 structural genes are present. Structural gene. This is the leader attenuated. This is the operator, this is the promoter, regulator and promoter and all. In this regulator, normally it is switched off because of RNA polymerase, then induces the repressor mRNA with the help of transcription process. That repressor mRNA Whenever repressor mRNA automatically it is synthesized, the translation process takes place, that is the aporepressor proteins are formed. This is aporepressor protein, that is nothing but tryptophan. Tryptophan protein is synthesized. Whenever tryptophan itself then unable to bind in that operator region, then automatically whenever aporepressor region automatically it activated. These structural genes are activated in this particular condition. Whenever activated, it is synthesized, the transcription and the synthesis of and formation of mRNA activated automatically to transcribe mRNA. Then in a translation process to synthesize polypeptide chain and all. To synthesize polypeptide, what type of polypeptides are formed? This is in this E enzyme as well as D enzyme, C enzyme and B enzyme and A enzyme. A group of polypeptide, these particular enzymes are formed. Here, that is raw material as well as enzymes, it formed tryptophan and all. Then automatically it must be formed tryptophan. Raw material along with this polypeptide, that means group of enzymes, it forms tryptophan. Here also tryptophan formed. Some amount of tryptophan, it acts as co-repressor. Tryptophan is formed co-repressor. Some amount of tryptophan acts as co-repressor and all. Then, what is the role of, already we said that lac-operon, operator, promoter or inducer or related regulator, their functions and all. What is the function of leader attenuated? It controls and slows down the action of polymerase. Whenever more concentration of tryptophan, whenever more concentration of protein, then automatically leader attenuated, one can slows down the action or combining of RNA polymerase. Automatically, it is also slows down the structural gene function, the structural gene functions, the transcription, translation, formation of polypeptide and all. Then this co-repressor. Similarly, here very simply you write this one like the same, and promoter, operator, leader, attenuated, then E, D, C, B, A. Leader attenuated, but where it locate leader attenuated between the structural as well as operator and all. Whenever it is formed, aporepressor, then if it bind with the co-repressor, whenever bind with co-repressor, then these particular, this is aporepressor, this is co-repressor. Whenever both are joined, then it acts as a repressor and ready to bind with the operator. Then inactivated or suppress or unable to transcribe or unable to synthesize, unable to translation, the specific transcription translation process stops whenever it binds. Whenever it binds. So here quite opposite. Their lactose, their earlier lac-operon, whenever lactose, it acts as that particular initiator and it acts as a inducer and all. Then automatically it becomes activated. Here, whenever additional tryptophan, co-repressor, whenever combined, it acts as a complete repressor. When our repressor bind on operator, then unable to transcribe as well as unable to translation, then formation of polypeptide. All these process it stops here. When our bind, that is important. Aporepressor itself unable to bind on operator. When our corepressor bind along with aporepressor, then it is ready to bind on operator, then automatically stop the further inactivation of the structural genes, then unable to transcription, translation process and all. So that is transcription. Similarly, histidine 9 structural genes are present. Like that, different types, it must be present related to this one. So in eukaryotic organisms, it is not at all represent any one specific gene control the protein synthesis, especially protein synthesis. Then who will be control these protein synthesis? You must remember once or twice. In prokaryotic only, operon, it acts as very important role, gene particular protein synthesis and all. Whereas eukaryotic organism, it is not at all involving particular genes and all. Then what it involves, what it control, the synthesis of the proteins and all. It involves all vitamins, depends upon the vitamins, depends upon the minerals, depends upon the hormones, that particular environmental factors. So all these factors control, regulate the protein or gene expression and all. So all these act as a vital role in this expression of the gene. That means the formation of protein synthesis and all. Protein synthesis means here formation of enzymes. Then what is the important role? Why? Because the gene expression act as a vital role. Whatever the embryo, that development and differentiation and formation, that means the control. When our embryo develop and differentiation, total it control and coordinate a group of that particular genes and all. There is no genes, there is no development, there is no differentiation, this particular embryo and all. So that much vital role takes place in the gene and all. Related to cancerous gene, it is also splitting genes related to here only in that eukaryotic formation of transcription, in the translation, the splicing process, splitting genes act as a vital role to separation of introns and the ligase which combines like this. The genes act as a crucial role in that particular organisms. Okay? Remaining will continue next class and all.